People trickling in. All right. Hello, everyone. Welcome to another session of our evolution seminar series. Today is about the wonderful world of fungi, as we're joined by Emile Gluck-Thaler, assistant professor, Department of Plant Pathology here at UW-Madison. He's come to talk to us about the fascinating topic of starships, giant fungal mobile genetic elements. So let's all give a warm welcome to Professor Gluck-Thaler.
Great. Thanks, Justin and Emily and Chris, for the invitation to speak with all of you today. So one of the most important motivators in my lab for doing science is that we know that understanding fungal threats and opportunities is really critical for safeguarding global food security and global public health. So fungi, of course, are the largest kingdom of microbial eukaryotes. You might have heard of them before in the context of yeast. So that's one type of fungus, but there are also a lot of other types of fungi that grow in filamentous forms. You might have also heard about mushrooms. That's another type of fungus. And something that is really fascinating about this kingdom of life is that they form very intimate associations with plants and animals. And really, these interactions that fungi have with plants and animals span the symbiotic continuum, right? So you have fungal pathogens that threaten global food supplies and also threaten human health and health of livestock. You have fungal commensalists who are just hanging out in our guts and hanging out in the guts of livestock, helping digest some food. And then you also have mutualists, which confer both benefits to their host in exchange for something in return. And I think it's precisely because fungi are in this position to intimately interact with plants and animals, this makes them key determinants of plant and animal health. And so if we want to understand how to safeguard and how to manage plant and animal health, it's really important for us to understand how fungi actually work. So what makes a pathogen, what makes a mutualist, and what potentially drives transitions between mutualists and pathogens. But there's a universal challenge to actually understanding how fungi work, and this is a universal challenge in organismal biology in general, and that's the fact that individuals within populations often differ very dramatically in their phenotypes, and so in this case, differ dramatically in their capacity to actually interact with their hosts. And so, of course, this is an evolution seminar series, so it's really important to consider this dimension of biodiversity, so dimension of phenotypic diversity within populations, because only traits that actually differ across individuals are capable of evolution, right, or capable of evolving. And not only are these traits capable of evolving, but really this variation from a practical standpoint complicates our ability to actually predict the outcomes of interactions that fungi are having with their hosts, and also makes it difficult for us to predict how fungi are going to respond to different treatments that we might use to actually manage those interactions with hosts. So if you think about in the context of a pathogenic interaction, if someone is infected with a fungal pathogen, you want to have a good idea of how that pathogen is going to respond to some of the antibiotic or antifungal therapies that you might want to use to cure that infection. And so the main point here is that if we can actually better predict the outcomes of interactions that fungi have with their hosts, predict the outcomes that fungi have to treatments that we use to manage those interactions, then we can actually be prepared to prevent and to manage pathogenic interactions and mutualistic interactions and to actually manage these threats and opportunities. And so the major question that my lab focuses on, broadly speaking, is what are the genetic bases of phenotypic variation in fungi? And how can we use this information to not only predict outcomes, both in the clinic and in the field, but how can we use this understanding to actually build and enrich our understanding of organismal evolution as a whole? And so the approach that my lab takes is three-pronged. So first, we start out with a computational biology approach. So here we develop workflows and develop new software, apply that software to big sequencing data sets, so usually transcriptomic data sets or genomic data sets. And we do this in order to discover novel patterns within those data sets. And then usually we'll follow that analysis up and interpret those patterns through the lens of evolutionary genomics. And really what this allows us to do is identify processes that are responsible for generating these patterns that we observe. And at the same time, we also generate some comparative insight into the evolutionary process as it's playing out across different organisms and species. And third, we try to follow all of this up with some molecular genetics approaches, which allow us to actually functionally characterize and functionally test some of the hypotheses that evolutionary genomics and computational biology are really so good at generating. We do this through both traditional molecular genetic approaches of knocking out genes, overexpressing genes, introducing genes to different backgrounds, and also by conducting manipulative experiments. And so over the past couple years, and hopefully for more years moving forward, we've been applying this approach to study an emerging phenomenon in fungal biology that is particularly relevant if we care about phenotypic variation. And that's the phenomenon of starships. And so, full disclaimer, these aren't starships that are at all involved in space travel. They're not the starships that are going to be taking humanity to Mars, apparently. But perhaps more relevant for this seminar series, what starships are, broadly speaking, is a newly discovered mechanism of fungal evolution. And so what does that mean? So specifically, a starship is a large transposable element. So you might have heard about this in some of your other classes that might have introduced this concept through Barbara McClintock's discovery of jumping genes in maize. So these are essentially genetic sequences that are capable of mobilizing and transposing between different locations in the genome. And in some cases, as we'll see a little bit later, they're also able of transposing themselves between the genomes of different individuals through a process called horizontal gene transfer. But what makes starships so unusual in the world of eukaryotic transposable elements is that they are 1 to 2 orders of magnitude larger than any previously described eukaryotic transposable elements. So they range between 20 to 700 kilobases in length, which is almost approaching the size of some smaller chromosomes in certain fungal species. So some really great work by my colleague and collaborator, Andrew, has identified just a single gene, which we refer to as the captain. This gene encodes a tyrosine recombinase that's both necessary and sufficient for moving the entire genomic region, which is actually quite remarkable. And then, yeah, as you might expect, in such a large chunk of DNA, there are a lot of other genes in addition to the captain that are carried as cargo by the starship. And this, of course, creates a lot of potential for some evolutionary shenanigans to emerge. So when we think about starships in the broader context of transposable elements, traditionally, some of the smaller transposable elements, when we're interested in understanding how they impact phenotypes, typically the effects of these smaller transposable elements are limited to the mutagenic effect of their insertion, right? So if they insert into a gene, they could potentially knock out function of that gene, or if they insert into a regulator, they could change the expression of that particular gene. So one cool example of this is the textbook peppered moth evolution, right? Textbook case of adaptation, where during the Industrial Revolution, moths went from a lighter phenotype to a darker phenotype to help them blend better into the background of trees that had pollution on them, right? And what's been kind of cool over the past couple of years, as more people have begun to study transposable elements in general, is that the actual genotype underlying this phenotype was found to be a transposable element that had inserted into the promoter of one of these genes that encoded for melanization. But typically, right, we're thinking about these transposable elements only in terms of their insertional effects. Some other transposable elements that are maybe a little bit larger are able to carry fragments of host genes, like helitrons or mavericks. And in general, this was kind of considered the upper limit of transposable element size for quite some time, right? And here, you're also getting some really cool phenotypes that are encoded by these transposable elements, but typically, because they're only encoding gene fragments or single genes, the types of phenotypes that can emerge from these transposons are still relatively quite limited. And then really what sets starships apart from all of these other transposable elements in the eukaryotic transposable element bestiary really is that they can carry tens and tens and sometimes even hundreds of different genes on them, right? And so you can imagine that this gives them a lot of potential to actually impact host phenotypes in often very complex ways. So what do we know so far about the transposable elements? So since we first described this superfamily just over 2 years ago, there's been a lot of research to try to understand like what they are, where can we find them, what are they doing? And so I'm just kind of summarizing all of that here in just a couple of lines. But as you can see here, so most of the references that I'm citing here are preprints. So it means they haven't been peer reviewed. Their manuscript has just been prepared and is in the process of being submitted or going under peer review. And yeah, as you can tell by the proportion of preprints to actual published manuscripts, we're still really just at the beginning of understanding what these elements are doing. And this is a really active field. So what we do know is that the Starship superfamily, you can divide it up into 11 distinct families. And each of those different families are differ from each other in terms of their sequences, but also in terms of where you find them across the fungal kingdom. The sequences are often really different from each other. Even closely related starships will often carry very different sets of genetic cargo. And although each of these families have their own taxonomic distribution, most of them are enriched in the Pizizomycotina. So these are a subphylum of fungi that contain thousands of species, many of which are involved or function as plant pathogens and animal pathogens. And most importantly, I guess for some members of the audience, these are filamentous fungi. So actually, we've never been able to find these elements in yeast-like fungi. But when you look back and you try to trace back when the last common ancestor of all these diverse sequences last existed, we can infer that last common ancestor existed around 400 million years ago. So this family of transposable elements has been diversifying for quite some time and has evolved to interact with fungal genomes in very diverse ways throughout all of that time. So getting back to this larger motivating question of what are the genetic bases of phenotypic variation, what we're going to do in the talk today is reframe this question through the lens of Starship biology and maybe rephrase it as a more testable question. Do starships actually generate selectable variation within fungal populations? And if they do, how are they doing it? And so the main takeaway from today's talk really is that starships fuel fungal evolution. And they do this in a couple of different ways. And so two of the projects that I'll be talking about today show how Starship activity generates variation in genome structure, also in the content of genomes, and also in the expression of genes within those genomes. And then I'll also talk about a project showing how starships really are these agents of horizontal gene transfer that can actually mediate the exchange of genetic information across species barriers, and in this way, create variation within different fungal populations. So we can jump right into this first project here, but before going ahead, does anyone have any questions? Does everyone kind of understand like what a mobile element is or what a transposable element is? Okay, seeing some nods, which is a good thing. All right, so this first project over here, essentially we took a tool that I had developed for finding starships and for annotating starships in fungal genomes. And kind of as a side note, before this tool, it was really difficult to identify starships. They don't have a lot of features that would make them really easy to identify a priori. And so we think that this is probably one of the reasons why they've successfully remained hidden for so long. So, so this tool, though, overcomes a couple of those bottlenecks and really allows you to systematically and in a high throughput way find where starships are in your particular genome of interest. So we took this tool, Starfish, and we applied it to a large population genomic data set from Aspergillus fumigatus, which is a globally distributed fungus. You can often find it in compost heaps, but it has the unfortunate distinction of also being able to grow in the lungs and in the tissues of immunocompromised patients. And so this fungus actually represents a pretty significant threat to human health at a global scale. So we applied it to this large data set. We sequenced a couple of new genomes that were a little bit higher quality than the rest of the sequences that we had. And yeah, this project was in collaboration with a couple of different labs from across the US and Europe. And so essentially what we did here, we used the tool, applied to the data set, and came up with an atlas of Starship diversity within this single fungal species. And then over the next couple of slides, I'll just show you how we interrogated that atlas to understand a little bit more about the biology of the elements. So really just to set a baseline, we found within this population of fungi, at least 20 distinct starships are actively transposing, right? And you can see them over here on the right. We've given them a bunch of different names over here so you can keep track of them. Maybe not through this presentation, but hopefully as you read the manuscript. And a couple of things jump out, well, should be jumping out right away. First is that these starships, each of them, have relatively patchy distributions across the population. So up here on top, we have just a phylogenetic tree describing the relationships between different individuals within this population. And you can see that there's a lot of presence-absence variation in where these starships are found. And sometimes you see there's actually quite some distance between the individuals, quite some phylogenetic distance between individuals that have a starship or don't have a starship. So that's what you see over here on the left in the heat map. On the right, we just have a summary of some alignments between the different sequences of these starships. And what I hope you can see here, I'm not sure if it's coming up in the back, but these little lines here connecting the sequences are showing regions of similarity between the elements. And basically, each of these elements are pretty diverse. They're carrying very diverse sequences that differ from each other. And in most cases, each of these 20 starships are carrying distinct combinations of genes that differ from other elements in the population. And then something interesting that we noticed is that if a starship is present in an individual, it's only ever present in a single copy, which is, again, slightly unusual when you might be thinking, like in regards to how you might be thinking about transposable elements as being like a high copy number and scattered across the genome. But the fact that they're only found in a single copy suggests that there might be some sort of mechanism controlling their copy number, although we don't really know what that mechanism might be. So where exactly are all of these starships hanging out in the genomes? Well, we took all of that data and then mapped it back onto the genomes themselves to find where the starships are and actually where all of these starship insertion sites are. And we found that the starships are generating structural variation really at a genome-wide scale. So over here we have a graphical representation of a genome from one of the individuals in the population. And in red, you can see all of the locations where we found a starship. So there are 8 different starships in this one individual. But then along the perimeter at each of these black tick marks along the circumference here, that indicates a segregating insertion site. So that means that some other individual in the population has a starship at that location, but this particular individual is missing a starship at that location, right? And so when you look at this, it's an indication that there's significant segregating variation within the population that's being generated through the activity and transposition of these particular elements. And so, yeah, we can do all sorts of statistics like add up, what percentage of the genome is starships, or we find close to 2.5% of any individual, right? Any individual's genome is going to be locked up into these starship elements. And we find that there are 154 sites distributed across the entire genome where you find evidence of a segregating insertion site. So really here, the take-home message is that really nowhere in the genome appears to be safe from the insertion of these particular elements. And just to give you like a sense of what this looks like, I pulled out one of those regions over here. Most regions just involve a clean insertion, but the dynamics in this particular region get a little bit wild. So over here in red, I've kind of boxed out all of the starship elements that are present. And you can ignore the other colors for now. But again, here we're just showing an alignment between different individuals. So sequences that are shared are going to be indicated with these gray links between the sequences. And what you can see really is that the starships are responsible for generating pretty significantly large indels or insertions and deletions across these different strains, right? Such that in the absence of these elements at this locus, right, the locus is around 400 kb in length, but at its maximum with all the starships inserting into it, in this one particular individual over here, the size of this region almost doubles, right? And we don't really understand what the significance of this is, right, for three-dimensional genome structure, or for the actual functioning of the genes within this particular region. But that's definitely something that we're actively pursuing and interested in following up on. One more thing to note is that that's kind of interesting from a population genetics perspective, is that most of those sites in the genome segregate at pretty low frequencies. And so what that means is that you're only going to find a couple of individuals in the population that have a Starship at a particular site, even though the frequency of the element itself could be much, much higher within the population. And so we're still not really quite sure how to interpret these results, right? The fact that you find pretty high frequency of Starships in the population, but then if you look at any particular site, it seems the frequency seems to be pretty low. But I think one way to interpret this is that beyond any like obvious deleterious effects of where the Starship is inserting, right? Because if you imagine if it inserts into an essential gene, then the individual with that insertion is not going to be doing too well, relatively speaking. But beyond any of these obviously deleterious effects, it may not actually matter phenotypically where a particular Starship is inserted in the genome. And so it can kind of hang out at a lot of different regions in the genome, with maybe no obvious consequences for either its own fitness or the fitness of the host genome at large. So after looking at where all these Starships are found at a genomic landscape level, we then wanted to take a step back and understand, okay, what are Starships doing at the level of the pangenome? So a genome, right, is just the set of genes within one individual, but a pangenome is the set of genes across all individuals within a population. And people are really interested in actually understanding diversity of pangenomes, not only of individual genomes, but of pangenomes, because this represents a more accurate depiction and a more accurate calculation of what is the genetic diversity within a particular species, right? And so when we put together the pangenome of Fumigatus, and then we just counted up how many of those genes are actually found on a Starship, we find around 3 to 5% of all genes in the species are found on Starships. And maybe more interestingly, around 10 to 16% of the accessory genes are found in Starships. Accessory genes are genes whose presence and absence differs across individuals within the population. These are really interesting from an ecological perspective because very often these accessory genes are involved in some sort of ecological interaction, be it an interaction with the host or interaction with other species or adaptation to a particular environmental factor. So this is maybe not too surprising when you think about what these elements actually are, right? They're mobile elements that can transpose, right? They could be easily gained, easily lost. So yeah, you might expect for accessory genes to be enriched on these elements relative to the rest of the genome. And I think what's interesting about this is that here we have significant amounts of variation within the species that's actually being organized and stored on these mobile elements. And just as like a comparative point, when you look to see, like relatively speaking, how much sequence is located on plasmids in bacteria. So plasmids are another type of mobile genetic element that resembles Starships in a couple of important ways. You see that plasmids are typically mobilizing around 25% of the pangenome, or 25 to 30% of the pangenome in bacterial species. So here, what we're finding is that Starships are mobilizing similar amounts of genetic sequence compared with plasmids in bacteria, which is kind of cool. So something interesting to note, so if you notice here, one of the criteria for defining, what genes in the pangenome are carried by Starship, we had this criteria that we had to find the gene at least one time, right? And the reason we had to make this criteria is that very often, not all closely related genes, not all closely related copies of a gene are going to be found on a Starship. So there's actually fluctuations in the strength of Starship to cargo associations, right? So to put this another way, there's some genes that are more Starship-y than others. So some of them seem to have these very strong associations with Starships. So if you ever find them in a genome, chances are they'll be on a Starship. Other genes tend to have weaker associations. So only occasionally will you find them on a Starship. Other times you'll find them in the rest of the genome. And that's kind of what these graphs here are summarizing. So on the y-axis over here, we have either the number of isolates with a particular gene or the number of isolates missing that particular gene. Each column over here is a gene that's either found in a particular starship over here on the left, or just summarized across all those 20 different starships in the population. And then the bars are color-coded about according to where we find that gene in a particular individual, right? So all these red bars over here indicate that this gene is actually found in this starship across, it looks like maybe 6 individuals. But then over here, it's actually not found in a starship in 1 or 2 other individuals, right? And so I pulled out these 2 examples here, right? Because you can kind of get a sense of, like, in TARDIS, for example, most of the genes on this starship are always found in a starship. But in Gnosis, there's only few of those genes that are ever really found on a starship, and they seem to be just hanging out in the rest of the genome. So this is telling us a couple of things. First, I think it's important to really understand, if you're interested in studying the evolution of a particular gene or gene family or phenotype, it's really important to understand the degree to which those genes are associated with starships, right? Is there a strong association? Is there a weak association? And I think by doing that, you can get a better sense of, like, what are the evolutionary dynamics that are impacting this particular phenotype. And so talking about phenotypes, right? This is all just a lot of data about genotypes, but how are starships actually impacting phenotypes within this particular population? And when working with Fumigatus, this fungus is actually a model organism, not only because it's a pathogen, but there's been a lot of work done characterizing genes in this particular species. And so we have the benefit of just cross-referencing all of our starship genes with large libraries of phenotypically characterized genes. And when we do that, we find a lot of really interesting cases of the starships carrying genes that contribute to adaptive phenotypes. So one of the best examples here is this one particular cluster that is called the hypoxia responsive morphology cluster. That's been really well characterized. It's been shown to increase growth under low oxygen conditions, which is particularly relevant during the infection of human tissues. And it's also been shown, probably as a consequence of that, to contribute to virulence in different strains. And the way that this cluster does that is by modulating the actual 3D architecture of the biofilms that this fungus forms when it's colonizing plant tissues. So what's kind of cool is that this cluster of genes and a couple of genes within the cluster are found across pretty diverse starships within the population. So these are the genes over here that have been phenotypically characterized. We don't really know what the functions of these other ones are, but presumably they're also involved in similar either biofilm-related processes or hypoxia-related processes. So that was one cool example. We also found another couple of examples of biosynthetic gene clusters encoded on these starships. So here are 2 examples that were actually initially identified around 6 years ago, or 7 years ago now, as being mobile within the Fumigatus species. So this means that these researchers found that the clusters were on different locations in the genome, and they hypothesized that perhaps it was due to some sort of transposable element activity. So over here, we were able to confirm that, yes, it is due to transposable element activity, and specifically due to the activity and mobilization of these starship elements. What's kind of cool is that since this first paper was published about these 2 mobile clusters, there's been some really nice phenotypic work actually characterizing the molecules that are produced by these gene clusters. And they're involved in some really interesting adaptive phenotypes. So fumahapeside over here, for example, this is a molecule that's involved in promoting thermotolerance and UV resistance in this particular species. And the molecule up here, fumagermin, this is a secondary metabolite that can actually is secreted by the fungus to suppress the germination of Streptomyces spores in the environment, presumably because this fungus, as it's growing in compost heaps, is competing with these bacteria for resources. So this is a kind of a cool way that the fungus is manipulating the developmental programs of its would-be competitors around it. So already these are some pretty good indications that starships are impacting important ecological traits in fungi. And we want to understand maybe a little bit more globally, like what's the bigger picture here of how Starship genes are behaving or being expressed under different conditions. And so what my collaborator Adrian did was he conducted a meta-analysis of RNA-seq expression data, right? So he was taking expression data from, I think, just over 20 different RNA-seq data sets and looking at the expression of Starship genes across different conditions, including like different nutrient manipulations or antifungal exposure or in-host infection. And he found a couple of different things. First, he found that the expression of the same Starship differs according to the environment that it's found in, which is perhaps maybe not that surprising, but that's indicative of a genotype by environment interaction, suggesting that particular genotypes of Starships are responding differently or being expressed differently depending on the environment that they're found in. But then more interestingly, in my opinion, he was also finding that the same Starship that's found in different individuals is expressed differently under the same conditions. And so that indicates that there's some sort of genotype by genotype interaction happening here, genotype of the Starship interacting with the genotype of the host genome, that's producing variation in a way that we might not necessarily have thought about before actually looking at the expression data, right? So it's not just enough that a Starship is present, you also have to take into account maybe epistatic interactions that are happening between that particular element and the genomic background. So that kind of wraps up the first part over here, just showing using this atlas of Starships from this one particular species, showing how Starships are fueling evolution at the level of creating variation in genome structure, genome content, and also gene expression. So the next project I wanted to talk about today is exploring a different aspect of Starship biology, and that's their role as agents of horizontal gene transfer. So over here, this was a project done in collaboration with Andrew and Aaron, who are currently working at Uppsala University in Sweden, looking to see how a set of Starships carrying a gene cluster involved in formaldehyde resistance has actually been horizontally transferred multiple times across different fungal species. Right, so the story begins here with the SSF, which is a gene cluster for degrading formaldehyde. So the cluster itself consists of around 6, sometimes 7 genes over here that each catalyze a step in this pathway over here for degrading formaldehyde. Over here, Andrew did some really great work just confirming the functions of these different genes. And actually, this was the first time a formaldehyde degradation pathway had ever been characterized in fungi. So that was kind of fun to explore. And we were, of course, initially interested in this particular cluster because we had found it on a Starship and no one had ever looked at formaldehyde degradation ever before. But then we all kind of got obsessed with understanding the role of formaldehyde in fungal ecology because we had never heard of it before. But apparently formaldehyde occurs naturally and is all around us. And particular different plants or plant tissues or plant products like fruit, for example, can actually accumulate formaldehyde in pretty high concentrations. And these can be toxic to the microbes that are growing on these plants. And so you can imagine that different environmental fungi or saprophytic fungi or fungi that are interacting with plants could be encountering pretty high concentrations of formaldehyde in their environment, not only as a consequence of human pollution, which is, I think that's initially how I was thinking about formaldehyde concentrations. But yeah, so like apples can have 6 times as much formaldehyde as jet exhaust in them, in parts per billion, which I think is kind of wild. So we were getting interested in this phenotype from an ecological perspective. We were interested in the cluster because we found it on a Starship and never been looked at before. But actually, as we continued to look into it, we found that not only does one Starship carry it, but actually 4 different Starships carry this SSF cluster. And all these 4 Starships here, so Typhon, Chrysaor, Mithridate, and Prometheus, again, here's one of those alignments where you can see that these elements really share very little sequence similarity to each other beyond this one cluster of genes over here. So the cluster of genes, you know, their identity ranges anywhere from around 60 to 99%, whereas there's just 0 identity, right, between any other parts of the elements themselves. So this suggests that this particular cluster is almost in a way being passed around like a hot potato between these distantly related starships. And so we wanted to understand a little bit more about the evolutionary dynamics behind this observation, what process could have led to this pattern. And so what we did was we built out a tree of the captain sequences of starships. So this is across all currently known starships over here. So in this way, we can kind of use the captain's evolution as a proxy for understanding the evolution of the element itself. And then we just look to see like how many of these captains or how many of these starships are associated with the genes in this cluster. And what we see very clearly are just like 4 spots in the evolutionary history of starships where the cluster has become associated with a starship element, right? Suggesting that these different events are actually independent of each other. We're not seeing inheritance through vertical descent, right? Instead, we're seeing different and repeated convergent evolution of this cluster becoming associated with different starships. And then what was kind of cool when we took a closer look at what species are carrying each of these starships, we found evidence that each of these independent acquisition events coincides with sometimes multiple horizontal transfers of the element itself, right? So Mithridate's been transferred between penicillium species and different Aspergillus species. Prometheus has been transferred between Aspergillus species and a Pathology species, and also the same over here for Chrysaor. And so essentially what this is meaning, what this is telling us is that the convergent acquisition of the cluster is really priming fungi for adaptation via horizontal gene transfer. And so one of the ways that this is kind of an aside, but one of the ways that we identified horizontal gene transfer is just by aligning the genomes of different fungi to each other and looking for parts of the genome that are more similar to each other than you would expect by chance. So that's essentially the graphs that you're seeing over here. So each of these dots is like a chunk of DNA sequence that's been aligned between one fungus to another. And you can see in most of the cases, because these fungi in these comparisons are pretty distantly related to each other, the bulk of sequences in their genome are only around 70% similar or identical to each other. But in red over here, you have all of the sequences in these particular starships, right? So what's kind of cool is that when you go and you align them, even though most of the genome is only 70% on average identical to each other, these starships are often the exact same sequence, right? They're not over, but around 100% identical to each other, right? And so you're always seeing them popping up in the top left-hand corner over here, suggesting also that these are all relatively recent horizontal transfers of these sequences across all these different starships and across all these different fungi, which is kind of cool. So of course, yeah, we want to understand what effect horizontal transfer might have on the recipient fungus. So we went in and phenotyped both a putative donor and a putative recipient. So over here, Aspergillus was predicted to have sent this starship over to Pathology over here. And in each case over here, we phenotyped multiple individuals within each of these species. And as expected, it was the individual with the starship indicated with the positive sign over here that was able to grow better on formaldehyde compared to other individuals in the population that didn't actually have this starship and didn't have the cluster. And what's kind of cool about this is that initially when we had started working on the project, we didn't have this Aspergillus strain, but we were just searching large sequence databases for fungi that could have this particular starship, and we found this Aspergillus strain, and then we actually ordered it. And it's a kind of a cool example of like a reverse ecology approach where you're able to at least generate a hypothesis about an organism's phenotype based on its genotype. And so as predicted, right, this particular Aspergillus fumigatus individual that has a starship is also able to grow better on formaldehyde. And yeah, this kind of really gets me excited because I think there are probably a lot more possibilities for predicting fungal phenotypes based on starship genotypes that we're really just beginning to understand. So before closing out this project, I wanted to just take maybe a little bit of a step back to understand the broader context in which starship activity is playing out during the course of fungal evolution. So here we have a phylogenetic tree that's summarizing the evolutionary history of the formaldehyde gene cluster specifically, right? So we're trying to understand how the different formaldehyde or how the cluster itself has been evolving across time. And when we build out this tree, what's kind of interesting, of course, we expected to find some of these sequences associated with starships, right? Because we know this cluster is found in starships. But what we didn't expect to find was that some of these sequences are actually not associated with starships at all. And other sequences are associated with regions of the genome that look like they once were starships, but are no longer starships because they've lost some of the machinery that actually enables them to transpose. And so what this is suggesting is that the broader picture here is a little bit more complex than just beyond just cargo being transposed by starships. But what this is suggesting is that the evolution of these sequences is actually intertwined with the starship life cycle of birth, movement, and death, such that the evolution of these sequences has really been marked by the fact that they've been picked up as cargo by these starship elements. So we see examples where cargo has become mobilized because it's been acquired by a starship, which can lead to horizontal transfer between different species. And then we also see examples here where the cargo, once again, kind of becomes part of the genome again once the starship dies, right? And we're really curious to understand what's happening along the way, right? So what are some of the consequences of being located in a starship, or potentially being located in a region that was a starship but no longer is, in terms of both the evolution of the sequences and then also the evolution of the phenotypes encoded by these particular sequences. So wrapping up the talk, and I'm going to wrap up the talk just by going back to this first motivating question. Are starships actually generating selectable variation? And if so, how are they doing it? So I've shown you guys some data from a couple of different projects on different fungal species, suggesting that starships really are fueling fungal evolution. And they're doing this by impacting genome structure, genome content, and also gene expression, and also acting as agents of horizontal gene transfer. And so I think all of these different projects are summarized by this model over here that we've put together to try and understand what exactly is the impact of this newly described mechanism of evolution. And so one way to think about fungal genomes is through the concept of different genome compartments. So a lot of different species have the genomes of a lot of different species have particular features, right, that can sometimes distinguish them from each other. So very often, if you're reading some comparative genomics literature, you'll see reference to either core genomic regions or dynamic genomic regions. And typically what this is referring to is just differences in gene density across these different genomic regions. So for example, core regions are typically considered to have pretty high gene densities, and the genes there are involved in more core functions that are really essential for organismal survival. In contrast to dynamic genomic regions, which are maybe a little bit more gene sparse, have more repetitive elements in them, and typically contain genes involved in ecological processes. And so what we're suggesting here with the starships is that they actually, in fungi at least, they represent their own distinct compartment that's really different from the rest of the fungal genome as described by a lot of these different models that we use to actually understand how fungi are evolving. So we're calling this the cargo compartment over here. And really, I think what this model is going to let us do is really help us understand how starship activity and how the starship life cycle intersects with the evolution of fungal genomes and the evolution of fungal phenotypes. So we've seen some examples about how being located in the cargo compartment, right, means that you can now be mobilized through starship activity. And some of the results of that mobilization is either moving to a new individual via horizontal gene transfer, or moving to a new site in the genome via transposition, and also potentially being lost. I didn't have a chance to get to show you some of these data, but you can imagine that not every transposition event is ultimately successful. Some actually fail in the sense that the element does not reintegrate back into the genome. So in this case, when that does happen, within just like a single mutational event, you can actually lose an entire starship. And that can happen, we think, in as much as 10% of transposition events. So the transposition is a little sloppy, but what that means is that these sequences can also be rapidly lost from a population, which again is another mechanism of generating variation. So once mobilized, these starships, of course, can continue on their journey of horizontal transfer or spreading to new genomic sites or being lost from populations. And then eventually, maybe if the selective conditions change, the starship can die, which actually results in the demobilization of the cargo. But by that point, that cargo might have already found itself in a new species or maybe found itself in a new genomic location, once again, generating some genetic variation that selection can then act upon. So what are some of the next steps here? All of that was really kind of phase one. So we know that starships are here. We know that they exist. And now the challenge is going to be to try and understand how their dynamics and how their activity acts to either facilitate or constrain the evolution of fungal phenotypes. And so at the beginning, I mentioned that, something unusual about starships is that they're carrying so many genes, and this gives them this increased capacity for actually interacting with their hosts in a number of different ways. So one perspective that I'm curious to explore is just thinking about the starships themselves as these like mini symbionts that are living within the genome, right? And depending on how they're interacting with the host, depending on the effect of the cargo that they're carrying, you might imagine that they're interacting more like a mutualist, right? In the case where they're providing a benefit and then also receiving a benefit in return. Other starships maybe are acting more like parasites by carrying cargo that promotes their own fitness, but at the expense of the host, right? So there's some examples of starships that carry meiotic gene drives, right? Which are definitely not good for the host, but good for the starship in the sense that it can promote its spread through a population. So I'm interested in exploring what different events drive transitions across this parasite to mutualist continuum. You might expect like a certain changes in the environment, for example, might make starship mutually beneficial in one condition, but perhaps more parasitic in nature under another environmental condition. And then, of course, beyond understanding some of these drivers of transitions, also just fundamentally want to understand the starship genotype to fungal phenotype map in a couple of different areas that are really important to consider when you're looking at and studying fungi, such as antifungal resistance and host pathogen interactions, and also lifestyle transitions when fungi move from being mutualists or commensalists to actually emerging pathogens. And so, yeah, with that, I'd like to thank everyone in the lab, Annika, Connor, and Samuel, who actually hadn't joined the lab yet when we took our first photo just a couple of months ago. And then also like to thank some of my collaborators here across Europe and the US, and happy to take any questions. So thanks. Right.
How exactly do these starships get from one species into
Yeah.
what even multiple conditions?
Yeah, that's a really great question. And Andrew and Aaron over here from Uppsala, they're actively pursuing that now to try and replicate it in the lab and trying to understand the different conditions that might promote or inhibit that.
So do they need to worry that they might jump in?
Yeah, that's an interesting question. I don't know. Yeah.
Given this part,
Right.
do you have any indication where it's
Yeah, that's a good question. So we think they're derived from their most recent common ancestor is another type of fungal transposable element called a krypton. Kryptons they use the same mechanism for replication, but they are always smaller and you never see any larger. So that's our best guess at where these things came from. They don't seem to be viral derived, at least most recently. But yeah, whether or not we can find them in other species, I don't know. We haven't looked. I will say we do find them sometimes in mushroom forming fungi, basidiomycetes. Yeah. And we don't actually think that they're active in the basidiomycetes, but rather they just ended up there because they were transferred in. So if that is true, then that would mean that they are able to expand, but maybe it's like a dead end host in the sense that they can't really replicate once they're there, but they can actually make it in. So, yeah. Oh, yeah. Yeah, well, so they're, as far as we know, they're just two essential features of a starship that actually allow it to transpose. And so the first is the captain, and the second are sets of conserved repeats at the ends of the element, and that those mark the boundaries of the element. So you need both of those things to be intact in order for transposition to occur. And if either of those are lost or mutated, then that kind of leads to the death. So yeah, it is really just kind of as simple as taking out the captain and yeah. Yes. Oh, yes. Yes, I will. Yes, for sure. Yes. Yeah.
So I'm interested in how spiders.
Yeah.
So they're massive. So I assume when they transpose out, it's really hard for the two streams to ligate back right way. So do you see like very weird structural variation for fungal species that starships?
So the question is, yeah, so the question is like, do we see structural variation in fungi that might be due to starships? And is that somehow related to how the starships are transposing and what secondary structures they might make during the course of transposition? Is that okay? Yeah, so we based on some comparisons to some other elements that also move via a tyrosine recombinase mediated mechanism, we think that the intermediate is actually circular in nature. So both of the ends will actually, I guess, ligate together during that initial excision event. And then the whole molecule or starship will move as like a single circular molecule to a new location. And then through site-specific recombination, will reintegrate into the genome. But we haven't seen any like particular like structural variation within fungi that might support or not support that hypothesis. I think we just kind of have to catch it at the moment when it is moving. Yeah, Justin.
So you mentioned that you found this general single copy.
Yeah.
As you mentioned, that's kind of so we need to understand about transposable elements. What do you think ultimately is it's functional chance? I think that you explained just by the physical constraints of the DNA molecule transpose. What are your thoughts?
Yeah, so the question is, why do we find starships only in single copy? And yeah, I guess what are some speculative thoughts about why that is? So there are a couple of interesting genomic defense mechanisms that fungi have to protect themselves from mobile element expansion and copy number increase. One of those mechanisms is called a repeat induced point mutation. And so essentially they're this molecular machinery in many fungal cells that scans the genome for duplicated sequences. And if it finds a duplicate sequence, it will mutagenize that sequence by basically increasing the AT content of the sequence. So we know that starships can be impacted by this RIP mechanism. So that potentially is one way that their copy number is being kept under control, just through interacting with fungal genomic defense mechanisms. But yeah, that's not really that satisfying an answer because they're, all transposable elements interact with RIP, and some are able to expand faster than RIP can actually keep them in check. So yeah, I think something else is also happening, and I'm not sure if it's host control or Starship encoded, for example. But yeah, now that we're developing some genetic tools where we can introduce starships or delete starships, I do want to just create some strains with multiple copies and actually just see what happens, right? And maybe it is due to the fact that, yeah, maybe, looking from the Starship's perspective, right? It might be actually beneficial to keep its copy number low, right? And so maybe a mechanism has evolved to control its own copy number because that would impose a lesser fitness cost on the host for carrying that much extra DNA or something like that. But yeah,
Related to that.
Yeah.
Here it is. You know, person can even make it or
right.
have anything to do. Just
Yeah. Yeah, that's a good question. That could also be an opportunity for starships to actually transfer between different genomes or between different nuclei. So maybe transfer is actually happening between, like, in a heterokaryon state, not necessarily between different individual mycelium, right? Or I guess that, yeah.
So we have a question in the chat that will read out. Could we identify gene coalitions if fungi provide insights into cell organisms and plants for animal genomes?
Yeah, that's a great question. So already the study of starships has been informed by different models that were first pioneered in bacteria. So bacteria have these really cool mobile elements called integrative and conjugative elements, or ICEs. And they're also an element whose movement is catalyzed by a tyrosine recombinase, but unrelated to these tyrosine recombinases. But essentially, for all intents and purposes, they appear analogous to starships in that they integrate into the genome and that they're these really large genomic regions. So certainly fungi have benefited from work that's been done in bacteria. But then, yeah, whether or not we can then use all this, combined fungal and bacterial knowledge for looking at plants and animals, I think that's an open question. And I think part of the reason why we didn't pay attention to starships, or we didn't know about starships for so long, is that we just didn't think that it was possible. That's a transposable element this large could exist in a eukaryote, right? So once we get over that conceptual barrier, right, I really hope that people are going to go for similar things in plants or similar things in different animals.
So you mentioned RIP.
Yeah.
Like have many other
Right.
Yeah. Widespread RNA. And most macromolecules don't have
Mm.
any of these.
Yeah.
Have a patchy distribution in them.
Yeah.
What is the thing that helps that?
Yeah.
And they should enter it and
Right. So the question was, why don't we find starships in Saccharomycotina yeasts, given that the yeasts often lack a lot of the genomic defense mechanisms that you find in other filamentous ascomycetes? Yeah, that's a great question, and I don't think we really have we definitely don't have a definitive answer to it, but I think it has to do with the actual morphology of the fungus. So the current hypothesis, although I did mention that, the starships could be transferred, via heterokaryons, the current hypothesis is that they're actually transferred between individuals through anastomosis, and the ability to actually move between different individuals through anastomosis is what makes them ultimately successful and able to survive. So maybe the fact that that is just not possible in yeast, that they might be more locked into an individual's genome as opposed to being able to spread across multiple individuals in a population. Maybe that's why they aren't ultimately successful. But yeah.
Psychology term. Can you give me a definition?
Right. So what is the definition of anastomosis? So anastomosis is when so the fungal body, we call that mycelium, and mycelium is made up of individual strands called hyphae. And when different hyphae form a mycelium, they're all kind of connected to each other. But then you can have this really cool thing where different mycelium, so presumably what are different individuals, can actually fuse together when their hyphae join. And that process of hyphae joining up is called anastomosis. So yeah, the fact that different, quote unquote, individuals can merge at least over like some part of their body, that might be really important, an important way that the starships are spreading through a population, right? Yeah. I don't know. We should throw a starship in some yeast and see what happens. So yeah. Great.
So good to have a professor.
Thank you.
Check my ass, we'll go over to 13.